Good evening, everybody. ನಮಸ್ಕಾರ ಎಲ್ಲರಿಗೂ. ಈ ದಿನ ನಮ್ಮೊಂದಿಗೆ ಡಾಕ್ಟರ್ ಗಾಯತ್ರಿ ದೇವರಾಜ್ ಮೇಡಮ್ ಅವರಿದ್ದಾರೆ. ಅವರು ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಆಫ್ ಸ್ಟಡೀಸ್ ಇನ್ ಮೈಕ್ರೋಬಯಾಲಜಿಲಿ ಪ್ರೊಫೆಸರ್ ಆಗಿದ್ದಾರೆ. ಈ ಹಿಂದೆ ರಿಜಿಸ್ಟ್ರಾರ್ ಸಹ ಆಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸಿದ್ದಾರೆ. ದಾವಣಗೆರೆ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಕಾರ್ಯನಿರ್ವಹಿಸ್ತಾ ಇದ್ದಾರೆ. Madam has a career spanning over more than 26 years of teaching, research, extension experience with societal orientation in all of it. She has published over 100 plus peer-reviewed research papers in national and international journals of repute, and her research area include probiotic for healthcare system and bioremediation. And Madam is the recipient of coveted Dr. Kalpana Chawla Young Scientist Award by Government of Karnataka. And as Dean of Science Faculty and Director IQAC, she has developed various programs and policies for academic excellence in the Davangere University. Dr. Gayathri Madam has played a pivotal role in streamlining different regulations, inception of seed money for young talented teachers and students. He has guided many PG and PhD students for their research. As a director, skill development, tendered to train and develop professionals equipped with the right skills to contribute to society at large. She believes in the creation and promotion of intellectual network of people whose commitment and drive turn them into change makers and leaders of tomorrow. She firmly believes in academic and research excellence as a strong driving force for rural upliftment along with clean, transparent administration. She aims to achieve highest credibility for the university by making it as an institution of par excellence. Her mantra for success is work is worship and reach the unreached through education as the mission. This is the brief overview of the academic and research experience of Madam. With this brief background, request Madam to deliver a talk on magnificent microbial world, a special emphasis on selected microorganisms. I request Madam to take over and deliver our talk. Over to you, Madam.
Thank you so much, Dr. Ramesh sir, and thanks for the opportunity provided. I take this opportunity as my privilege to talk to young, talented students of +2, and because this age group is very volatile, and what is good for their future is just they are either their parents are skeptical or our children or students are very much immature to decide. Conventional pattern of career would be like either going for engineering or medical, or taking up BSc as a basic course, something that of that sort. I would like to throw light in my talk with regard to the microorganisms that means too small to be seen by naked eye, and this unseen organisms can create havoc world over that everyone has experienced in recent times during the pandemic COVID world over. And we have also experienced first wave with little knowledge, second wave with more drastic with high knowledge, and also down the line it was a little bit less virulent during third wave. And we are also now hearing the news from China side that again the fourth wave or the transformed virulent form of COVID virus is emerging again. So this unseen microbial world is something fascinating. Therefore, our students of +2 must focus in this line, whether it is engineering or it is medical or whatever the field they choose. Their microorganisms exist either for the betterment of the existing basic process. Now we shall move on to the topic of today's discussion. Microorganisms are present everywhere, especially they are the native of water as well as soil, and transiently they are present in air. As air does not contain any nutrients, the organisms exist in air are temporary, either whipping up of the soil or the air droplet. The organism exists temporarily, and then with the gravity, with the density, the organism settle down during a period of time in the daytime or at a night time. However, through air, varieties of microbial diseases and allergy are caused through our breath. What is microorganisms? Microorganisms are too small to be seen by our naked eye. That means the size of the organism is below zero point 0.1 millimeter, and those organisms which cannot be seen by our own eye can be grouped as microorganisms. To visualize them, they requires microscope, especially compound microscope with a minimum magnification of 100 times is necessary to identify to observe the organism. As I have told you that the organisms are present in water, soil, but temporarily they are present even in air. Actually, the microorganisms houses in the body of animal, plant or human abundantly. Especially if you take up the microbial load on the surface of human, it is quite abundant. They are present in the gastrointestinal tract, alimentary canal, in the stomach. The pH of the stomach is about 1, where the number of organisms are minimum, especially those organisms which are acidic. That means they can able to tolerate the acidic concentration pH 1 can survive. If they can survive at that particular pH, then they can produce ulcers in the stomach. For example, you must have heard of a special organism called Helicobacter pylori, which can cause stomach ulcers. Likewise, as the alimentary canal descends down, that means small intestine to larger intestine, the pH gradually increases from progressively acidic to alkaline, and therefore the alkaline condition is ideally good for the existence of maximum number of organisms. In total microorganisms that are present in the body or restricted to gastrointestinal system, they can be called microbiome. Most commonly, five groups of organisms that we can come across in microorganism, like bacteria, virus, fungi, which includes yeast and molds, and then protozoa. Definition of the phrase microbiology is derived from Greek word micro means too small, and bios, as you are aware of, life, and logos means study. It's actually an interdisciplinary science which deals with the study of unicellular as well as multicellular microscopic organisms. The sub-disciplines of major microbiology includes bacteriology, virology, then mycology, that means study of fungi, then parasitology and immunology are other parts of other fields of study. From your 8th standard, you would be aware that microscope is one particular optical instrument that is designed to visualize the smaller objects to maximize, that means enlargement. Therefore, you will see the organisms the much stretched, and therefore visualization is brought out with resolution. That means distinct points that the two objects can be observed as a distinct point. So here microscope that was invented long before, and until the invention of proper compound microscope, visualization of microorganisms was not done thoroughly. If you go down the line, that how the history of how the microbiology as a special discipline has developed, much before Christ, Aristotle, who postulated that the existence of microorganisms, because we will not see that the microorganisms are existing in air or soil or in water. However, these organisms can cause disease or bring transformation in the soil, or they can decay the compost, or they can produce the compost, they degrade the debris. Likewise, they can cause the achlorotic patches on the surface of the plants. That means although they are seen as non-existing, but their debilitated or drastic ill effects are visualized. Likewise, it was predicted much before Christ. And during 1658, Athenaeus Kircher, who defined that the invisible organisms found in decaying bodies, they are also found in meat, milk, and even the secretion of worms in which the organisms are present. In addition to that, it was for the first time Robert Hooke who actually first to use the microscope to visualize the cell, and he described first to describe about the microorganism, and he drew the diagram of microorganism by pencil sketch and was published in Microphagia. You must have heard of a well-known scientist. Actually, he was called father of microbiology, Anthony von Leeuwenhoek, who actually had no formal university or college education. He was only a textile merchant, a draper, a Dutch. He had a hobby of grinding glasses and then making it compressed and observing through it. He has prepared a fixed single lens which can able to enlarge the image of the microorganism or the object to about 300 times. So his microscopes, that means 100 times, and he has prepared some 300 microscopes by using these lenses. And he observed various ovary of a plant and blood cells through his microscope, and even the content that is present in between the teeth are been collected and placed on a slide, placed on the surface of the needle, and showed that there are some small tiny animals in your mouth. There were many women in his house, and they scared that the mouth carried so much organisms. They were so surprised, and in a letter it says that Leeuwenhoek writes a letter saying that there are more, and he recites these small animals as animalcules. There are more animalcules in one person's mouth than that of the men in the whole kingdom. Likewise, the organism's abundance is too high. There was a controversy earlier that how the new smaller animals or smaller living beings are generated. They are either reproduced or automatically produced. Many had a concept that the organisms have smaller organisms develops on their own. But what is referred as abiogenesis, that means spontaneous generation of new organisms. While that is one school of thought, one group of scientists or a person's philosopher, whatever you call. While another group says that it is not a spontaneous generation, it is through biogenesis. That means through the fusion of two gametes, male and female, there produces the younger one, and therefore the generation of new ones takes place. While there was a fight between abiogenesis group and biogenesis group, and Francis Reddy, who actually showed that maggots develop only on the surface of the decaying matter where it contains organic infusion, where the organisms, when maggots laid the eggs, from that the new maggots were developed. But the John Needham was a supporter of spontaneous generation. While another scientist, Lazarus Spallanzani, he showed that boiling broth, that means when you cook meat or the vegetable, there develops a large amount of molecules, and the fluid will be dense, what you call broth, which gives rise to microscopic life, or a smaller organisms would develop. For example, you just cook vegetables and then remove the vegetables and keep that water as such, which contains large number of nutrients. If it is boiled and it is properly sealed, then no generation of microscopic forms takes place. So this particular explanation forms a foundation for the development of food preservation. Now what we get, all cooked food or the processed food that are available in the market, which is processed, which is sterilized. So repeated boiling that has resulted in the inactivation of lot of microorganisms. That means the organisms would not recover, and therefore the organisms that the food remains unspoiled for a long period of time. During 1895, Louis Pasteur, who proposed pasteurization effect, that means temperature that is employed at about 62 degrees Celsius, 62.8 degrees Celsius for a period of 30 minutes, where if the milk or the beer or other beverages are heated, majority of mesophilic organisms or spoilage causing organisms would be killed, and therefore further immediately it is cooled and then can be kept safe for consumption. Louis Pasteur, though he was a chemist, he has done quite an amount of work, and his period can be called golden era, golden age of microbial world discovery. And he has worked out on rabies vaccine in preparation of neurotoxins, neurologically effect causing rabies virus vaccine for rabbit dog bite. The individual will get the rabies disease, for which he has first produced the vaccine, and then he has identified the reason for a silk moth disease by a protozoa, and he has produced a good amount of vaccines against cholera bacilli, which would cause poultry infection. Later, John Tyndall, he actually proposed a type of heat treatment, what is referred as Tyndallization, a discontinuous method of heat treatment where at a temperature of about 100 degrees Celsius, the organisms that the food contained with the organisms would be killed. And later on, if the food was incubated, some of the organisms which are resistant could multiply upon incubation time. Discontinuous heating that has resulted in killing of organisms to the level of 100%. Another important discovery in the later stage was the use of phenol as a substance, chemical substance, in bringing about sterilization. A surgeon, namely Lord Joseph Lister, he was named as father of antiseptic surgery because earlier to that, operation surgery was used to be done while it was often failure due to the sepsis. That means pus would be produced and then the sutures would not be cleared and the individual would die, although the operation was success. Inspired by Louis Pasteur, where air contained microorganisms, this concept was deduced by Pasteur. He thought that some amount of phenol, if at all it is disinfected, if the room is cleared with the chemicals and the organisms, if at all they are killed, then sepsis can be avoided, pus production could be avoided. So he was successful in conducting such a program, and then he was therefore named as father of antiseptic surgery. And father of modern microbiology, this particular title actually goes to Robert Koch, who's actually a physician, and he has proposed 4 important postulates, that means hypothesis, which is actually true, and even it is now it is used in the laboratory to check the virulence of the organisms. That includes the organism should be present in all cases of disease, and when the individuals microorganism should be must be associated with a particular type of disease, and then these organisms can be isolated from the infected individuals and could be cultured. That is the second postulate. And third postulate is once the organisms are isolated, If it is injected to the experimental animal, then again the same type of clinical symptoms must be produced in the recipient. It may be a experimental animal or a human himself. While fourth postulate include, once again from the experimental individual, the organisms can be re-isolated and could be cultured. Now the fifth postulate that has come, that means the each of the organisms that are causing the infection must produce antibody corresponding to it in the system. So he has studied much about Bacillus anthracis, which produces anthrax, basically causing infection to animals and transmissible to human beings also. Another important discovery in the field of microbiology is development of a special type of chemical substance, chemical composition, what is called agar. This agar is actually a solidifying agent. One of the Koch's co-worker's wife co-worker was Fanny Hisse. His wife was Fra Hisse. She was using this agar that is collected from the seaweed red algae, and she used to purify it and used in dishes to make liquid into solid like halwa. And once when Koch visited his student's co-worker's wife, house, she prepared some dish which was very interesting, and the same agar he noted and then he used it in his laboratory, and for the first time solid culture media was developed, and now also we use in our laboratory for the purpose. And later that particular solid media must be placed on some dish. This is Richard Petri. Now we call that glass material as Petri dish. His invention now also we use for the growth of organism on the solid media on which the organisms like bacteria, fungus or whatever the protozoal even can be grown on solid media. And you are well aware that Edward Jenner, who actually for the first time developed vaccines against smallpox. In his home, he had observed some of the cow mates who had blisters on the in between the fingers, and then he extracted those blisters and then used as immunizing material. And later on, many of the individuals who were dying, we were suffering with the smallpox, he used this cowpox pustule preparation, and that was preventing the smallpox infection. And you know very well that now the smallpox since 1984 or so, this has become extinct now. There is no such cases are found because of the production of effective vaccines. While another scientist, Alexander Fleming, who discovered the antibiotic penicillin from the Penicillium notatum, now it is called Penicillium chrysogenum. And phenomenal immunological quantitative methods of estimation of antibodies was done by Paul Ehrlich. He studied about toxins, and corresponding antibody produced was referred as antitoxins. You see here the two pictures of Anthony von Leeuwenhoek and Robert Koch. Actually, the initial period, the abundant contribution of them resulted in the profound development, later development of microbiology. And at the center, you can see the primitive microscope with the crude lenses, and then a small needle. You can see here that the sample was mounted here, and a fine one hole you can see here where the eye must be placed, and then the fixed organisms or the sample materials must be observed through it. And this laid a foundation to revise, to make many, to implement many mathematical principles, and therefore we can develop some compound microscope in the later days. I hope you have in your biology book 5 kingdom classification of living organisms such as Monera, Protista, fungi, plantae and animalia, in which if the organisms are divided into 2 groups based on their nuclear material, prokaryotes and eukaryotes. Eukaryotes are unicellular organisms, or it may be multicellular organisms. Unicellular organisms They include Protista, for example, fungi that includes yeast and molds, while the multicellular organisms with cell wall or without cell wall, Animalia or fungi and plantae, while Monera includes eubacteria and archibacteria. And you can see here the eubacteria are classed as heterotrophic or autotrophic, and archibacteria have three groups or more than three groups: halophiles, thermophiles, and methanogens. In salty environment, these organisms are living, and then in hot spring, in acidic environment, these acidophilic organism grows, and methanogenic organisms grows on marshy areas. Autotrophic bacteria can be of two groups, like they can synthesize their own food through chemical means, what is referred as chemoautotropes, or through the usage of light. Some of the organisms can able to synthesize their food, for example, cyanobacteria. In general, if you classify the organisms as two groups like cellular organisms and acellular organisms, prokaryotes and eukaryotes are the cellular organisms. They possess a typical cell which lack a true membrane enclosed nucleus. For example, bacteria, general bacteria or archaea are in this group. They have distinctly located with their genetic material within a false or a pseudo membrane sometimes, or they are like nuclei but not nuclei. Oid refers to like nucleoid. As you are aware that eukaryotes are the organisms which contain a distinct, well-defined nucleus and has membrane-bound organelles, for example, fungi, animals, and plants. Acellular organism groups are those particular microorganisms which has no typical cell structure or no enzymatic energy production in their system. They merely consist of genome like DNA or RNA or a protein code. Generally, this protein code is derived from the host cell. For example, viruses or viroids or prions are included in this group. The domain of life that contains prokaryotic cells which has distinct cell wall, and the cell wall is made up of a complex biomolecule, what is referred as peptidoglycan, and these bacteria that possess a typical rRNA composition. Some of the examples, generic examples of bacteria include Staphylococcus, Streptococcus, Lactobacillus, or Escherichia coli, and many more. While archaea, this bacteria, they do not have peptidoglycan in their cell wall. They are the primitive group of bacteria. They have typical lipids in their cell membrane, and they have archaeal rRNA in them. For example, halophiles or methanogens or thermophiles. To represent fungi, fungi, the fungal cell, it is an eukaryotic organism which has membrane-bound organelles, clearly defined nuclei. There are about more than 1,44,000 known species of organisms in the kingdom of fungi, and unicellular fungi is yeast, and multicellular fungus is known as molds. And these organisms, especially molds, causes rust, smuts, mildews, and some of the beneficial molds includes mushrooms. Another group of unicellular filamentous bacteria is actinomycetes. They are gram-positive organisms which measures about 1 to 2 micrometer, very small organisms, but live in the soil, and they have abundant economical or environmental interest. They produce varieties of antibiotics. Example, like some of the species of actinomycetes includes Streptomyces gracious. You must have heard of streptomycin produced by this organism, Streptomyces venezuelae, and Micromonospora purpurea, and some variant of the Streptomyces nocardia and nocardia species. Another group of organism, what is referred as algae, the members of this group is predominantly aquatic and photosynthetic organisms. They have many types of life cycles, and the size of them ranges from micromonas, which is very small, very few micrometers in size, to as large as the giant kelps, which reach to about 60 meters in length. Number of microscopic algae, which include chlorella, cyanobacteria, spirulina, volvox, eulothrix, and many more. And just now we have discussed about the virus, which consists typically of nucleic acid of their own, and they have a protein coat. They are just like any other materials like soil or the sand or a book on your study table. But when it finds a suitable host, it starts multiplying and it takes control of the host cell cytoplasm totally, and they either rupture the cell, even they can enter into bacteria, and bacteria can be lysed, or even fungal cell, to which can, virus can enter and they can break, and from one particular cell they can produce about 10 to the power of 7 infectious viral particles out of it. Or in the animal cell, when the organism enters to the human body or animal cells, they enter into the cell cytoplasm and they cause infection, and they migrate to the healthy cell, and then they infect the surrounding cell and bring about a pathological effect. You are well aware that microorganisms are omnipresent. They are present everywhere. If you take up the human body, that the existence of microorganisms are distributed throughout the body, starting from the head to toe. On the surface of the skin, the organisms are localized, and these organisms species and the diversity is something marvelous. For example, the skin microbiota, which includes the Staphylococcus aureus or Staphylococcus epidermidis, or some of the organisms like diphtheroids or Haemophilus influenza or pneumococci, are present on the surface of the skin. And while if you take up the respiratory system, we have good number of systems that are existing in the human body, like respiratory system, gastrointestinal system, in which the abundance of microorganism is too high, while in the central nervous system there are no organisms are present. If at all organisms are present as a normal microbiota, like any other like respiratory tract or a gastrointestinal system, the central nervous system breaks down. That means a high 100% protection is afforded by the human system not to enter any of the organism into the brain. If at all they cross the barrier, then individuals succumb to the infection and the total collapse of the system takes place. We breathe through our nose, nostrils, and then it enters into the oral cavity, and then alveolar. Down the line, it is through the tongue, and then it will reaches to the trachea, and then the lungs, and then breathing takes place, pumping takes place. You know, a well organ, the architecture of the nose itself is so nicely designed, such that the organisms that are present in the air are filtered or deposited over the nose surface, and a fine cilia that is present in the nostrils will make, which is so sensitive, which propels the entry, or it catches the microbial entry deep into the respiratory system. If the organism, for example, the size of the organisms or the materials, if at all it is over 10 micrometers, then it will be caught by the fine ciliary thin hair in the nostrils. If it has been escaped, then there are the sensitive mucous bed which collects the, which covers the microorganisms which are about 2 to 10 micrometer of size, and immediately cough reflex takes place. And during cough reflex, number of microorganisms are released out of the respiratory system, and you should be aware that during such cough reflex, there is a release of drops, isn't it? So this drops which evaporates or they bifurcate to become droplets, and then droplets will be dried with the water content, and then the sodium chloride that is present in the droplet, which makes the droplet into droplet nuclei, which measures a size of about 10 micrometer. So this is what is referred as infective molecule, which when enters into the person who is healthy, uh, when they breathe, when they inhale, that can infect, that carries the microorganism, can cause infection to the healthy susceptible. So normal microorganisms in the normal microbiota, what we call, they are present in the respiratory system. Upper respiratory system includes Staphylococcus aureus, Staphylococcus epidermidis, then Haemophilus influenza may be present, or some of the species of Streptococcus. In addition to that, in the mouth cavity, Staphylococcus aureus or Neisseria, actinomycetes, lactobacillus, and some form of yeast are also present. In the intestine, as I have told you that in the stomach, the pH is very low. That means it is highly acidic, pH of about 1. The number of organisms are too low. As the gastrointestinal system descends down, the pH increases, and therefore the total load of microorganisms increases. At the distal or the terminal part of the intestine, the number is about 10 to the power of 13 per square centimeter area of the intestine would be present. Remember, students, it is the organisms can multiply, one can become 2 in a span of about 18 to 20 minutes, and overnight time it can grow, one cell can grow into 10 to the power of 7 to 13 cell, and such high number organisms are grown, and then it must be put out. The defecation, that means the stool that would contain about 25% of the organisms would be 25% of the solid material would contain only microorganisms in them. Therefore, the safe defecation is too important. Otherwise, these organisms can cause infections to some other who are susceptible. Likewise, the organisms that are present in our system in different area are diverse, and if the organisms are misplaced from one area to another, then this can cause altered effects. Sometimes it may not cause any effect. Sometimes it can be more violent and produce various types of diseases. Therefore, maintenance of personal hygienic condition as well as environmental safety is too important. You know, when you take bath, when we take bath, large number of organisms are washed away, and then they will be removed, and then these organisms, the organisms as such, they are called native microbiota or normal microbiota of the system multiply, and then they build up the population. And even if you don't work, don't do any activity, at the rate of 10 to the power of 2 to 3, that means 100 to 1000 microorganisms are released to the environment continuously, and therefore we would be constantly polluting the environment even. And then this is one astonishing feature, and therefore the dissemination of the organism takes place. Some of the microorganisms that are important in various fields include like agriculture, environment, medical, food and dairy, and industrially important organisms, and then pharmaceutical important organism. And microorganisms are also used in biotechnological applications, like they are also useful in molecular biology and environmental engineering. Let's focus, I'm going to highlight only few of the examples, like the microorganisms that are present in the soil, which are having importance in plant growth promotion or soil fertility, and also they would help in they will initiate the plant diseases, and also they can help in promoting the promoted as a biocontrol agent. One of the important organism in the soil environment is a rhizobium. It is associated with the formation of root nodules, and they live with the root system in a symbiotic way. That means in close association, they help in production of typical type of nodules. As an example of Mimosa pudica, or even in groundnut arachis hypogea, and they have a typical enzyme, what is referred as nitrogenase enzyme. In an anaerobic environment, they fix the atmospheric nitrogen. You are well aware that the atmosphere contains about 78% of nitrogen, inert nitrogen, needs to be fixed for production of proteins, and therefore the plant growth takes place. And close association of these rhizobial species, some are symbiotic, some are non-symbiotic, they fix up nitrogen, and therefore they help in promotion of the plant growth. And on the Petri plate, how do they appear? When you crush the root nodules and then place it on a typical yeast extract mannitol agar media, they form a mucoid colony like this. You can see this to make one unseen bacteria, to make it visualize like this individual colony. It requires about a population size of about 10 to the power of 7 cells to produce such a colony. Another important organism is trichoderma, which is fungi that reduces the occurrence of plant disease, and they compete with the soil pathogens, and they synthesize some of the antifungal metabolites, and then they kill some of the pathogenic organism by producing lytic enzymes and degrade the cell wall of competing plant root pathogens, and therefore they are widely used as a biocontrol agent. Another symbiotic association example is Azolla F1 and the Anabena algae association. In the symbiotic association also, Anabena fixes the nitrogen on the leaf surface of Azolla, and therefore this nitrogen is supplied to the host plant, and then even this combination is employed in the fish culture as well as in fed to the cow in producing high amount of milk, and it is an enriched food for the nutrition. Special organisms in the environment. Environment includes the soil, air, or the water environment. You know that through air, the inhalation, we inhale air, and then sometimes through cough while talking or sneezing. For example, some of the organisms like Mycobacterium tuberculosis, which if an individual is suffering from TB, tuberculosis, when he coughs, sneezes, or even talks, how the organisms can enter into the respiratory tract of the healthy susceptible individual, and then it can cause the infection in the recipient, can be visualized here. So one of the important organism to cause such a disease is Mycobacterium tuberculosis. It was one of the rampant disease that is occurring, affecting the individuals with cough, sneeze, and then reduced body weight, and then the fever discontinuous, and it can even cause central nervous system disorder also. It is highly fatal, for which to prevent this particular disease, a vaccine that is developed, what is referred as BCG vaccine, Bacillus guerin and Comet and guerin, Bacillus, Comet and guerin, by using mutated or less pathogenic strain of Mycobacterium tuberculosis on the media for a long period, that is for 13 years, it is grown to make it less virulent, and that particular material is used for the production of vaccine. Then another important airborne infection is through the inhalation of rhinovirus that will be present in air through the infected individual, through the sneeze or cough, it will be entered to the healthy susceptible individual. If an individual is affected with TB, sputum examination is done from the by using a swab or from the sputum cough. The sample is collected and then it is stained through a special staining technique, what is referred as acid fast staining, because though it is a gram positive organism, it is not a simple organism. It possesses high amount of a waxy substance on its cell wall, and therefore special staining technique is necessary to identify. In an infected individual, you can see a bacilli which is in a joint like a chain like arrangement. In another slide, you can see here as an individual cell which is in the form of branch. When the individual is highly infected, the organism's density is also will be high. Another important organism of our interest is Escherichia coli. It is an intestinal organism. Actually, it is an aerobic organism, or we can call facultative anaerobic organism, but it can grow aerobically also. The organism is a friendly organism which lives as a normal natural microbiota of the intestine. It is a beneficial organism commonly called coliform. But the same organism, when it is released to the environment, when it is contaminated to ponds, lakes or streams, it can produce, when it is consumed, it can produce serious effects to the consumer who consumes it. The major infectious disease that is taking place with the Escherichia coli infection is urinary tract infection and diarrhea. There are, this is the morphology of Escherichia coli cell under the microscope, and then some of the typical media that is used for the cultivation of this organism that is called Mekonkes media. You can see tiny light pink colored colonies which are indicated as lactose fermenter. On eosin methylene blue agar, you can see a jet black colored organism with a metallic sheen here. The organisms also cause the infection like urinary tract infection or diarrhea. Diarrheic infection caused by this organism is of typical interest. There are about 5 different diarrheic group of Escherichia coli, like enteropathogenic Escherichia coli, enterotoxigenic or enteroinvasive, enterohemorrhagic or enteroaggregative. So study of this organism is of 2 interest, because one is it is a very friendly organism. It helps in digestion or improvement of the individuals by producing, defending against the pathogenic organism in the intestine by producing a typical antimicrobial product, what is called colicin. While the same organism can become pathogenic and can cause urinary tract infection or varieties of diarrheic infection in young children or the adults or aged ones. And it can also produce pyogenic infection and septicemia. The organism is also called indicator organism, because if the organism is present in water, that says that it is an organic pollution or a fecal pollution. But the organisms can live in the water, especially in drinking water, for a period of only 7 days time, 8 days time, a week's time. That says that the organisms, if it is detected, then it says that it is a recent pollution. So it is called indicator organism for the organic pollution of the drinking water. Another organism that we have just seen, Vibrio cholerae. It's also a gram-negative, a facultative anaerobic coma-shaped organisms. They are originated from the marine environment and adapted to even freshwater environment, and they produce typical choleric infection as a severe form of diarrhea and dehydration. If at all not been treated, then it becomes fatal. Individual may die within few hours. It produces a rice water type of diarrheic stools, and individual will suffer severely, and then the total discharge of the water content from the body is the twice the weight of the individual. The organisms will not do anything, but it leads to hemoconcentration and cardiac arrest because of the increase in the viscosity of the blood. Then base deficit acidosis, the body feels it is highly acidic. Then another group of organism that is of interest here is algal blooms in water sources. Pseudoniscia species of algae And some forms of diatoms, they commonly cause red tides. Especially these red tides are seen in the coast of California. Harmful algal blooms that develops like this. You can see how the sea is appearing too red, and the aquatic animals dies off because of the production of the toxin, because they consume such a toxic algae. And even it's not directly fatal or dangerous to human beings, but it can be fatal if at all person who swims and swallows water. And an interesting thing here is some of the bioluminescent algae, that especially Noctiluca, and they are often called sea sparkle. They are small in size and make the entire sea as bioluminescent. And therefore, the sea at that particular area in Caribbean island, it is called bioluminescent bay. The sea sparkle is so abundant as that of neon blue lamp at night. And then in a drop of water, the number of this Nautiluca or the bioluminescent algae is too high. About 1,000 to about 1 million such small cells can be observed. Another important bioluminescent mushroom you can see here, it is of two interest how the luminescence of the mushroom can be seen. There are more than 80 species of bioluminescent mushrooms are recorded. Artimillaria melea are the more widespread bioluminescent fungi in the Northern America, and Mycena species, it's called eternal light mushroom across the rainforest of the Brazil can be seen. And you know that when first time the rains, immediately you smell something earthy, and this is called the smell geosmin. That is due to a metabolite produced by Streptomyces gracious, a volatile isoprenoid compound responsible for the earthy taint or the smell, what is called geosmin. So this Actomyces, especially Streptomyces gracious, is abundantly present in the soil. Some of the medically important mushrooms, like Taiwanese mushrooms, namely Anthrodia and Anthrodia species, Anthrodia camphorate and Anthrodia salmonia, they possess high anti-cancerous activity. Especially it was tested against breast cancer cells. And remember, it costs so high. That means per kg, it costs about 80,000 rupees. And then another thing is that it is used to change the skin color from black to fair. And the Ganoderma, another Ganoderma lucidum, also called the Lingzimo mushroom, it cost 7,000 rupees per kg because it possesses a gut bacteria, and this is highly lipolytic, and they can break the lipid content, oil content, and helps in weight loss and prevent obesity. The organisms that are present in the extreme environment are called extremophiles. For example, thermophiles, acidophiles, barophiles, that means live in the high pressure environment, and also the methanogens. Hot springs and the Yellowstone National Park that contains the organisms of hot springs. Especially a thermophile is an organism that tries to live at higher temperature between 41 to 122. Therefore, at a high temperature enzymatic transformation, a organism called Thermus aquaticus is used. Another group of organisms is called methanogens. This is a diverse group of organisms. They can produce methane as a major product of their metabolism. This methane gas generation is too important in producing energy. Rumen is the cow or the buffalo which feeds which grazes the grass and which enters to the stomach. The ruminants are possessing a double stomach, and then they the organisms, the plant materials are being digested. Bacterial digestion takes place to produce hydrogen and carbon dioxide, and then later methanogens produce methane output. Varieties of organisms, Methanococcus, Methanogenium, and Methanopyrus, and the Methanosarcina species, Methanospera, are the important organisms in this line. To your interest, microorganisms are having a normal microbiota that are present in the system, helping the individuals normally. But these organisms, some of the organisms, they can be too dangerous. They can be used in bioterrorism also, in biological attack. It may be intentional release of virus, bacteria, and other germs can cause, they can kill or sicken people and livestock or crops. For example, for in this line, Bacillus anthracis that causes anthrax are used. Sometimes virus. Most dangerous pattern of organisms can be classed like this. Category C is the influenza virus, and then B is the salmonella, and the category A is most dangerous one is the smallpox and anthrax virus. Microorganisms are too important in food industry because microorganisms can help to ferment and increase the value of the foodstuff, or they can even cause a spoilage, or food can be the disease causing agent where the organisms can produce the disease, and through the mechanical carrier they can produce the infection, or sometimes the microorganism can produce toxins in them and then produce the intoxications. In this line, lactic acid bacteria is one of the important beneficial organism, and they help in fermenting the milk or other products to substances to transform to produce, for example, sourdough bread production, or cheese production, yogurt production, or a special cabbage fermented food, what's referred as sauerkrauts, pickles, beer, wine, and other fermented foods and animals like silage. The organisms, the lactic acid bacteria, produce large amount of lactic acid that could be used as food preservatives. And as a nutraceutical, probiotics, you must have heard of this terminology, which promotes the health of the individual, and as medicines or the supplements as a growth enhancer, this is being used. You can see the picture, that is normal fermentation of the dosa batter that we commonly use for either idli or dosa, which is fermented majorly by lactic acid bacteria, and they bring about health benefits to the consumer. And this particular slide shows you that in our laboratory, we undertake the research by using varieties of species of lactobacillus, and their potentiality would be has been checked for the health promotion. Like we have checked for the control of colorectal cancer in the mice system by using lactobacillus fermentum or lactobacillus plantarum in combination, and therefore maximum level of control to the level of 80% has been seen with the mice system. In the same way, the probiotic lactic acid bacteria are helpful in hydrolyzing the gluten, and this help to improve the gut health, and in the management of celiac disease, that is that the food has been developed, wheat based food has been developed to control the celiac disease. Similarly, lactobacillus fermentum, another species of this, efficiently detoxified aflatoxin, a dangerous toxin produced by Aspergillus flavus, which produces liver intoxication and liver disease, even liver cancer, hepatocellular carcinoma is produced. Some of the potential lactic acid bacteria could able to detoxify, and in presence of chlorophyll, the detoxification process has enhanced. Similarly, the species of lactobacillus plantarum, fermentum, and pentosus and others could able to alleviate, that means control lactose intolerance and improve the gastrointestinal disorders in people who are intolerant or allergic to lactose. Likewise, the yeast also represent a beneficial effect in producing fermented beverages and in production of bread, that is in the daily use. They are the unicellular eukaryotic organism, undergoes budding like this, and then asexually they belong to Ascomycetes group, and they are helpful in number of ways when used in optimum level. Otherwise, overusage or over fermentation resulting in soaring. In addition to that, the production of vinegar, citric acid, and these organisms are used, and then sometimes the organisms can also cause spoilage, like you can see here. The overgrowth of this resulted in the increased keeping time resulted in the growth of molds. Here green colored fungus that you can see, aspergillus. Like food, the algae can be used as food here, like spirulina tablets are used commercially, especially by astronauts and others with the enriched protein content. While you must have heard during COVID-19 pandemic, spirulina chicky have been produced by CFTRI, which has boosted the immunological level. Resistance power of the individual has increased. In the medical sector also, the majorly the organisms, microorganisms can cause the disease. And as a microbiologist, it is the responsibility to detect the microbial infection and that result in the treatment and further prevention of the disease. Majority of the microorganisms themselves can produce the disease. The production of the disease is harmful effect to the individual, while at the same time, the same organisms can be used in the preparation of diagnostic kit and also used for the preparation of vaccines. Some of the highly potent organisms capable of producing toxins include Clostridium botulinum and Clostridium tetani. Especially the Clostridium tetani, which produces the neurological disorder. Neurotoxins are produced, what is referred as tetanolysin. More than that, tetanospasmin, which generate muscle rigidity. So neurological disorder include the paralysis. So another organism is Clostridium botulinum, which produces most potent toxin, more than hydrogen bomb we can call, because at a very small concentration they can produce gastrointestinal paralysis when consumed the food contained with the Clostridium botulinum toxin referred as botulin. Some of the capsule producing organisms are also dangerous, which resist the phagocytosis process, and then therefore they can be infectious. For example, pneumococcus, which streptococcus pneumoniae, which produces pneumonia. You must see this particular slide. Tooth decaying bacteria mainly involved organism here is a Streptococcus mutans. They produce abundant amount of acids and they act upon the tooth surface, especially the enamel, and forms and the disintegration of the enamel takes place, and therefore there forms a cavity. If the tooth is affected by this particular acid environment, then individual feels the sensitivity, and later on the organism burrows, moves deeper, and then the root of the tooth will be highly infected. If later also, if it has not been treated, then the organism enters through the blood portals and cause dangerous infection. Tooth loss would be one thing, and also it can cause other surrounding infection like the inflammation of the gum, and then ear pain will be there, and then down the line it can spread to the entire body. Candida albicans is another organism which causes oral thrush. You can see the tongue here. This is the Candida albicans, which is a yeast, pathogenic yeast that can cause infection to young child, a milking child, and the mother may suffer from the coated tongue with the infection. You must have heard of this organism, what is called black fungus. Post-COVID infection, there was a mucormycosis that has resulted due to the low immunity among the individuals. So mucor is one type typical organism which is actually can cause the infection to the eyes or the soft mucocutaneous tissues and produce such a blackened infection. Then coming to another representative is uh, rabies virus, which is causing rabies through the bite of the rabid dog. And um, we have discussed earlier that Louis Pasteur, who actually collected neurological tissues of the sheep and used that particular preparation as vaccine to inject to the individual who are susceptible for the infection. Coronavirus has caused greater news in the recent past 2-3 years, and then it is actually a old virus which has three distinct group of disease causing virus, like severe acute respiratory syndrome disease causing organism, then middle east respiratory syndrome organism, and even common cold causing organism. It has a special corona, like the spikes on its surface, therefore the corona word has been kept. Now the vaccines have been developed and it is seen its efficacy, and in India, you know that almost 100% vaccination is done. Therefore, we can combat against the coronavirus, although there is some mutation and new forms are generating. The ancient virus we can call the polio virus, which belong to the enterovirus group, a smallest RNA virus, that is picornaviridae group. They cause abnormality like the paralysis. Sometimes it is too dangerous, but once affected, it will cause residual effect in the affected individuals, and then the individual will not be suffered. Another group of organism here is the Rebiola or the rubella virus, RNA virus. They produce such forms of clinical infections, which is a contagious infectious disease in causing infection to the younger age children, and if proper immunity level is not developed, then it may recover in the adulthood. Organisms are also important in bioremediation, in remediating, in detoxifying the toxic substances in the environment, and therefore less toxic substances are able to be produced. So in our laboratory, we have used Stenotrophomonas organisms, and then the Pseudomonas can cleave the most toxic polychlorinated biphenyls to lead to the catechol production, and therefore the less toxigenic substances are produced. Therefore, they are helpful in bioremediation. So industrial applications of the microorganisms are quite abundant. They are helpful in producing varieties of vaccines. Therefore, we can protect ourselves from the forthcoming disease without suffering, or industrially important enzymes are produced, then organic acids are produced. So varieties of antibiotics are produced by good number of organisms which are having commercial importance. Vitamins and even cosmetics are produced. Then varieties of organisms can able to produce preservatives, and natural preservation can be done as generally recognized as safe for consumption. Varieties of vaccines that are produced by using microorganisms, which may include inactivated killed vaccines or live attenuated, that means crippled organisms, then mRNA vaccines, subunit vaccines, toxoid vaccines or viral vector vaccines, DNA vaccines or edible vaccines. Microorganisms are also too important in genetic engineering, especially Escherichia coli that are used in recombinant DNA technology. Primarily, they are used as vectors, and the hosts in they are expressed in the host gene transmission. For the expression, desired genes could be placed within a vector. For example, you can see human cell. We can cut out, we can take a small piece of DNA representing insulin gene, and then we can ligate, we can place it within a plasmid, and now it is a vector, and then this plasmid can be placed in the bacterial cell. Now the bacterial cell capable of producing abundant amount of insulin in them. So we can use that further for injection. we can grow abundantly, purify it and extract it, and can be injected to the people who are suffering from diabetes. Similarly, microorganisms can be modified by using genetic engineering techniques, and therefore the BT cotton, BT brinjal, golden rice, likewise several types of transgenic microbes can be produced. So these are the applications of microorganisms, and I have just thrown the light that various fields of microorganisms, various fields of study, and microorganisms applications. Therefore, how microorganisms can be used for benefit of the human health. The same organisms, if at all they are not maintained in the safer level, they can cause decomposition, they can produce disease, debilitation. So microorganisms are a real friend, because starting from the birth, the organisms are start accommodating before the development of even the immunological system, and they're progressively increased in their number, and upon the death also the organisms are going to decompose, and therefore microorganisms are a good friend in protecting ourselves against the infection in the system, and at the same time if we don't maintain them they can cause harmful effects.
How many slides are there?
Madam, namaskara. Hello. Hello.
Yes, sir.
Madam.
Yes, sir.
Thank you very much for a wonderful presentation. Right
Thanks.
from the basics, beginning of the bacteriology, Athanasis, culture in the start madidre, covered everything right from bacteria, virus, algae, fungus, and even food poisoning and uses of beneficial microorganisms like lactic acid, bacteria, and yeast. I'm sure you have covered everything that comes under PO syllabus.
Yes, sir.
I hope students will be highly benefited. So, madam, students have joined both on Cisco WebEx platform and YouTube. Some of them have joined on Cisco WebEx, and many would have joined in YouTube. And
Yes,
your presentation will be put on our website, which many
yes,
of
sir.
the students can see even later. I'm
Yes, sir.
sure students will be highly benefited for their this particular talk will be highly beneficial for them, because many of times we see few lecturers will not be able to uh, so eloquently talk about microorganisms like you. Students, I'm sure, would have taken a lot of take-home message from your talk. On behalf of KSTA and all the students present here, we are very, very grateful to you. Thank you very much for this wonderful talk. Mr. Umesh, are there any questions?
No, sir, there are no questions as of now, but we have requested them to post if any in future.
Madam, the students will be posting the questions later, and we will be forwarding it to you. You
Sure,
may
sir.
reply later.
Yes,
And
sir.
in
I'll
the
be
meantime,
more
we
than
have
happy to answer.
two questions for students to answer. Two questions based on your talk, which students will be answering, and will be forwarding it to you. You have to select two best answers out of
Yes, sir.
many that may reply.
The questions have been posted in the chat box, sir. Madam has given 3 questions. They have posted. Thank you, sir.
We'll forward the answers to you, madam. Please select two best.
Yes, sir.
In future also, KS will be organizing many such webinars. We'll be inviting you for talks. Please
Yes, sir.
accept and deliver a talk like this. We'll be inviting Devaraj also for future
Yes, sir.
webinars. Please
Yes,
convey
sir.
my regards to.
Yes, sir. Definitely.
Thank you.
Thank you so much, sir. Thank you. Thank you.
Thank you very much, madam. I'll be
Thank you,
sharing the
Umesh.
YouTube link to you, madam.
Yes, sir. Yes, sir. Thank you. Thank you.
Thank you very much.